I'm Isabella Bilecki. I'm a fourth year PhD student here at the University of Adelaide, and I work under my supervisor, Associate Professor Renee Turner. So I work here as a part of the Translational Neuropathology Lab, and my project is looking at improving patient outcomes post-stroke and what age does to a stroke individual. So a bit of background as to who I am and how I got here. I was really always interested in the brain and the things that can go wrong with the brain, being such a delicate little organ that we have in our body. And in year 12, I actually did a research project on Alzheimer's disease and found it really, really fascinating. So I did an undergraduate in health and medical sciences, which I really found that I loved the neuroscience aspect of everything that I was learning. And I was fortunate enough at the end of that degree that there was an honours project available in stroke and functional outcome post-stroke. So I took up that opportunity, and I'm now 5 years later towards the end of my PhD looking at how the brain changes with age post-stroke.
And can you tell us how that project's going?
So the project itself, I'm looking at how age first and foremost impacts the blood vessels that we have in our brain, as obviously they're the ones that are being damaged in a stroke, but what the environment around those blood vessels look like both in healthy aging and post-stroke. So I'm looking at what stroke at different ages looks like in the brain and how the environment around the blood vessels change. So in particular with my work, I'm focusing on what we call the blood-brain barrier. So the blood-brain barrier is a really strongly permeable barrier that really regulates what moves in and out of the brain environment from the bloodstream. So usually it's really tightly regulated so that we don't let anything just cross into the brain tissue. As I'm sure you can appreciate, it's a really delicate environment. However, post-injury, such as something like stroke, that blood-brain barrier starts to break down and we start to get larger molecules moving in and out of the bloodstream into the brain tissue. And namely lots of what we call neuroinflammatory cells, which I'll let one of my other PhD students talk to you about, that can create a lot of destruction within the brain tissue following stroke, which often results in that sort of worsened outcome for the individual.
So can you tell us a little bit about your experience doing honours and any advice you have for anyone considering doing the same?
Absolutely. I think the biggest piece of advice I could give is to find a project that really, really interests you. There is no point doing work that you're not going to love, because there will be times in the year where you sort of sit there and question why you are here. But from my experience, when I got towards the end of my undergrad, I actually had two honours projects on the table, two very different honours projects on the table. One which I ended up taking, which was the stroke research project, but another one that was in forensic anthropology. And I think in the end, the thing that got me across the line was I got to sit down with both potential supervisors and get to know them a little bit better, and I decided to continue on with the stroke project and work under the amazing Associate Professor Renee Turner and at the time Anna Leonard as well. So both of them were really, really critical, I think, in making that decision, helping me make that decision. It is really, really important to get to know the people that you will be working with. It is a very intense year doing honours, and you work with them day in, day out, even outside of your normal working hours, as I very quickly learnt. So it's really important to have a really good group of people around you, and I was incredibly lucky to have a fantastic support network around me when I did my honours year to keep me afloat through all those really, really tough sections of work.
So can you tell us about what sort of techniques you use in your research?
Yep, so lots of different things. So as I mentioned before, we looking at brain tissue, blood serum and CSF. So there's lots of pipetting that goes on, so if you haven't done that already in your undergrad, I'm sure you will at some point. And we use these microtomes, which are these really sort of precise, almost like a deli slicer type machine that really finally allows us to cut tissue in thin sections down to 5 microns. So we often sit there for hours on end sort of cutting tissue, using those that we can then use for staining tissue in IHC, so immunohistochemistry or immunofluorescence, depending on what we want to look at, which is where we get to look at specific proteins that we find in cells or expressed in the cellular environment itself. Outside of that, we also in our lab run lots of things like Western blot and ELISA that allow us to look for sort of quantities of proteins in the tissue and how much might be apparent in either brain tissue or in the serum, blood serum or cerebrospinal fluid as well. So outside of that, we look at lots of motor tests, cognitive tests and behavioural tests to see what sort of outcomes we see post-stroke. So we might look at things like working memory, we might look at things like anxiety and depression type measures, as well as sort of those functional motor assessments where we look at sort of sensory motor function as well as sort of limb movement. On top of that though, I've been really fortunate in that we've had the opportunity to look at some to organise some MRIs and look at MRI of the pre and post-stroke brain, as well as the MRIs of brains of aged brains as such as well, so we can have a look at sort of what's happening within the brain, not just in protein and cellular work, but sort of more macroscopically look at the size of the ventricles and where the infarct actually is within the brain and how it's progressing and impacting other areas of the brain.
And do you have a favourite instrument that you like to use?
I absolutely love sitting in microtoming because you can sit there with my AirPods in and I can be listening to a podcast while I do it. It's really rhythmic to sort of just sit there and cut the tissue for lots and lots of hours. I also really love embedding tissue, which is when we sort of get a brain sample and we process it and then put it in wax so that it can be kept sort of indefinitely. So sitting there and putting the little tissue samples into wax is really nice because it's nice and toasty and warm when you sit there and do it. So they're probably my favourite two things. When I can get on the microtome, it's quite a popular piece of equipment in our lab.
So can you tell us a little bit about what a typical day is like in the lab?
I'm going to say no 2 days are exactly the same. Some days you'll have days where you'll be doing the same thing, but nothing is always the same as the day before. So I can do anything from sort of collecting data, generating data, down to analysis of data. So in particular, we look at brain tissue, we look at blood serum, we look at cerebrospinal fluid as well, and at what protein and markers we can find in there. So my day-to-day could be anything from sort of cutting tissue really, really fine in order to stain and look for certain proteins or look for certain cell types within the brain tissue and see either where they're aggregating or how many of them are there in a particular region of the brain. It might be that I'm sort of pipetting out blood samples, again, to look for different protein markers and what levels and elevations we're seeing within the tissue at either different times post-stroke or at different ages that a stroke's occurred. And then I also look at sort of functional outcome as well, so looking at how a stroke impacts things like motor impairment, cognitive impairment and behavioural tests as well. So I often run a suite of different functional assessments that we can look to find out how, not only what's happening in the brain, but how that actually manifests in the sort of outcome that we see in someone post-stroke.
And what do you like doing when you're not busy here in the lab?
I'm always here busy in the lab most of the time, and if I'm not busy here, I'm busy at home doing some work. But no, outside of my PhD work, you'll often find me either on my yoga mat or curled up on the couch with a good book and a good glass of wine, or out with friends. So yeah, but honestly, it's mainly here in the lab.
Well, that sounds great. Thank you so much for being with us here today.
Thank you.